The amazing world inside. What is cell biology? Cell biology is a very interesting part of science. It is the study of cells. Cells are the tiny building blocks of all living things. Think of them like tiny bricks that make up a big house. Every plant, every animal, and even you are made of cells. These cells are so small that we usually need a microscope to see them clearly. Studying cells helps us understand how life works at its most basic level. It is like looking at the smallest parts to understand the whole machine. This knowledge is very powerful and useful for many things. Understanding cells is important for many reasons. When we know how cells work, we can understand how our bodies grow. We can also learn how our bodies repair themselves when we get a cut or a scrape. Cell biology also helps us understand what happens when things go wrong, like when people get sick. Many diseases start with problems in the cells. So, by studying cells, scientists can find new ways to treat these diseases. It is a field that is constantly discovering new and exciting things about life itself. This makes it a vital area of research. Cell biology looks at many different parts of the cell. It studies the cell's skin, called the cell membrane. It also studies what is inside the cell, like the cell's control center, called the nucleus. The nucleus holds all the instructions for the cell, like a tiny computer. Scientists also study how cells get energy, how they make new parts, and how they talk to each other. All these processes are very important for the cell to live and do its job. Each part has a special role to play in the life of the cell. Learning about cell biology can open up a whole new world. It shows us the incredible complexity that exists even in the smallest parts of living things. It reminds us that life is a very organized and detailed process. From a single cell to a large animal, the basic principles of cell life are often very similar. This study helps us appreciate the connections between all living organisms on Earth. It truly is a journey into the very essence of what it means to be alive. That we learn so much from these tiny structures. The building blocks of life. Understanding cells. Cells are the fundamental units of all known living organisms. This means that everything that is alive is made up of one or more cells. Some living things, like bacteria, are just one single cell. Other living things, like people or big trees, are made of many billions of cells working together. Each cell is like a tiny factory. It has many different parts that do different jobs. These parts work together to keep the cell alive and functioning. Understanding these basic units is key to understanding all of biology and life itself. Most cells have a few main parts. There is an outer layer called the cell membrane. This membrane acts like a skin. It controls what goes into the cell and what comes out. Inside the cell membrane is a jelly-like substance called cytoplasm. Many important cell activities happen in the cytoplasm. Then there is often a special part called the nucleus. The nucleus is like the cell's brain or control center. It contains the cell's instructions, which are stored in a molecule called DNA. These instructions tell the cell what to do and how to grow. The DNA inside the nucleus is very important. It carries all the genetic information for the organism. Think of DNA as a very long instruction book. This book tells the cell how to build proteins and other molecules it needs. It also tells the cell when to grow and when to make new cells. When a cell divides to make new cells, it must copy its DNA very carefully. This ensures that the new cells get the correct set of instructions. This careful copying is essential for life to continue correctly. Cells come in many different shapes and sizes. For example, nerve cells in your brain are long and thin. They help send messages quickly. Muscle cells are designed to contract and help you move. Plant cells have a strong outer wall that gives them shape and support. Even though they look different and do different jobs, most cells share the same basic machinery. They all need to take in nutrients, produce energy, and get rid of waste. And very importantly, they all need to be able to make more cells. This process is called cell division. Making more of you. An introduction to cell division. Cell division is one of the most important processes in all of life. It is how cells make more cells. Imagine one cell carefully splitting into two new cells. This is the basic idea of cell division. This process does not happen randomly. It is a very controlled and organized series of steps. The original cell is often called the parent cell. The new cells that are made are called daughter cells. These daughter cells can then grow and, in many cases, divide again. This cycle of growth and division keeps life going. Why do cells need to divide? There are several very important reasons. One big reason is for growth. When a baby grows into an adult, it happens because cells are dividing and making more cells. Your body gets bigger because you have more cells, not because your cells get much larger. Another key reason is for repair. If you cut your skin, cells near the cut will divide to make new skin cells. This helps to heal the wound. Cell division replaces old or damaged cells throughout your life. There are two main types of cell division. The first type is called mitosis. Mitosis is the process that your body uses for growth and repair. When a cell divides by mitosis, it makes two new cells that are exactly like the parent cell. They have the same DNA and the same number of chromosomes. This is important so that all your body cells have the correct set of instructions. Think of it like making a perfect copy of a book. Every page is the same in the new copy. The second main type of cell division is called meiosis. Meiosis is a special kind of cell division. It is used to make special cells called sex cells. In humans, these are sperm cells in males and egg cells in females. Sex cells are different because they only have half the amount of DNA as regular body cells. This is very important for sexual reproduction. When a sperm cell and an egg cell join, they create a new organism with a full set of DNA, half from each parent. We will look at both mitosis and meiosis more closely. Mitosis. Part 1. Getting ready to divide, prophase and metaphase. Mitosis is the process our body cells use to make exact copies of themselves. This is vital for growing bigger and for healing when we get hurt. Before mitosis can even begin, the cell has to prepare. This preparation phase is called interphase. During interphase, the cell grows bigger. It also makes a complete copy of all its DNA. The DNA is found in structures called chromosomes. So before mitosis starts, each chromosome has an identical copy of itself attached, like two twins holding hands. This preparation is crucial for successful division. The first actual stage of mitosis is called prophase. During prophase, several important things happen inside the cell. The copied DNA, which was long and stringy, starts to condense. This means it coils up very tightly and becomes visible as distinct chromosomes. Each chromosome looks like an X shape because it consists of the original chromosome and its identical copy, called sister chromatids. Also during prophase, the membrane around the nucleus starts to break down. This allows the chromosomes to eventually move around the cell. Special fibers also begin to form, which will help move the chromosomes. The next stage of mitosis is called metaphase. The word meta often means middle, and that is a good clue for this stage. During metaphase, all the chromosomes line up in a single file line right across the middle of the cell. Imagine soldiers lining up for inspection. This line is sometimes called the metaphase plate. The special fibers that started forming in prophase, called spindle fibers, are now fully formed. These fibers attach to the center part of each chromosome, called the centromere. One fiber from one side of the cell attaches to one chromatid, and a fiber from the other side attaches to the other chromatid. It is very important that the chromosomes line up perfectly in the middle during metaphase. This careful arrangement ensures that when the cell divides, each new cell will get exactly one copy of each chromosome. If the chromosomes do not line up correctly, the new cells might get too many or too few chromosomes. This can cause serious problems for the cells and for the organism. So, metaphase is like a critical checkpoint. The cell makes sure everything is perfectly aligned before it moves on to the next step of splitting the chromosomes apart. This precision is a hallmark of cellular processes. Section 5, Mitosis, Part 2, Splitting apart, Anaphase and Telophase. After the chromosomes are perfectly lined up in metaphase, the cell is ready for the next stage. This stage is called Anaphase. During Anaphase, the action really begins. The sister chromatids that make up each X-shaped chromosome are pulled apart. Remember, these sister chromatids are identical copies of DNA. The spindle fibers, which are attached to the chromatids, start to shorten. As they shorten, they pull the sister chromatids away from each other. One chromatid from each pair moves towards one end of the cell, and the other chromatid moves towards the opposite end. This movement in anaphase is very precise. The cell works like a tiny machine, carefully separating the genetic information. Once the sister chromatids separate, they are now considered individual chromosomes. So for a short time, the cell has double the normal number of chromosomes, but they are moving to opposite poles. This separation ensures that each new cell that will be formed will receive a complete and identical set of chromosomes. It's like carefully dividing a set of paired instruction manuals, so each new office gets one full set. This step is rapid, but very organized. The next stage, following Anaphase, is called Telophase. In Telophase, the separated chromosomes have reached the opposite ends, or poles, of the cell. Now the cell starts to reverse some of the changes that happened in Prophase. A new nuclear membrane begins to form around each set of chromosomes at each pole. This creates two new nuclei within the single elongated cell. The chromosomes also start to uncoil and become less condensed. They go back to their more stringy form, ready to direct the activities of the new cells. The spindle fibers also begin to disappear. Finally, the last step of cell division often overlaps with telophase. This step is called cytokinesis. Cyto means cell and kinesis means movement or division. So cytokinesis is the actual physical splitting of the parent cell's cytoplasm into two separate daughter cells. In animal cells, the cell membrane pinches inward in the middle, like tightening a drawstring on a bag. This continues until the cell divides completely into two. In plant cells, a new cell wall forms down the middle. The result of mitosis and cytokinesis is two new daughter cells. These cells are genetically identical to the parent cell and to each other. Section 6, Special Delivery, the Magic of Meiosis. Now, let's talk about another very important type of cell division called meiosis. Meiosis is different from mitosis in some key ways. While mitosis happens in most of your body cells for growth and repair, meiosis has a very specific purpose. Meiosis is the process used to make special cells called gametes. Gametes are sex cells. In humans, the male gametes are sperm cells, and the female gametes are egg cells. In plants, pollen contains male gametes, and ovules contain female gametes. These cells are essential for sexual reproduction. The most important thing to know about meiosis is that it reduces the number of chromosomes in the gametes by half. Our regular body cells, like skin cells or muscle cells, are called diploid cells. This means they have two sets of chromosomes, one set from each parent. For humans, this means our body cells have 46 chromosomes, arranged in 23 pairs. However, gametes are haploid cells. This means they have only one set of chromosomes. So, human sperm and egg cells each have only 23 chromosomes, not 46. This reduction is absolutely vital. Why is it so important for gametes to have only half the number of chromosomes? Think about what happens during fertilization. Fertilization is when a sperm cell joins with an egg cell to start a new life. If the sperm had 46 chromosomes and the egg also had 46 chromosomes, the new baby would have 92 chromosomes. That is too many. The baby would not develop properly. By having meiosis reduce the chromosome number to 23 in sperm and 23 in egg when they combine, the new baby gets the correct total of 46 chromosomes. Half come from the mother and half come from the father. Meiosis is a more complex process than mitosis. It involves two separate rounds of cell division. These are called meiosis I and meiosis II. Before meiosis I begins, the cell goes through an interphase. Just like before mitosis, the DNA is copied, so each chromosome consists of two sister chromatids. However, the way the chromosomes behave and separate in meiosis I is very different from mitosis. This difference is key to reducing the chromosome number and also to creating genetic variation, which means differences between individuals. We will explore these steps next. Section 7, Meiosis in Action, the two-step dance. Meiosis I is the first round of division in meiosis, and it is quite unique. It starts with Prophase I. During Prophase I, something very special happens that does not occur in mitosis. Homologous chromosomes pair up. Homologous chromosomes are the pairs of chromosomes you get from your parents, one from your mother and one from your father in each pair. They carry genes for the same traits. These pairs find each other and stick together. While they are paired up, they can exchange small pieces of DNA. This process is called crossing over. Crossing over shuffles the genetic information, creating new combinations of genes. Next, in metaphase Y, these paired homologous chromosomes line up in the middle of the cell. This is different from mitosis metaphase, where individual chromosomes line up. In metaphase I, it is the pairs that line up. Then in anaphase I, the homologous chromosomes in each pair are pulled apart. One chromosome from each pair goes to one pole of the cell, and the other chromosome goes to the other pole. Importantly, the sister chromatids are still attached to each other. This is a major difference from anaphase in mitosis, where sister chromatids separate. Telophase 1 and cytokinesis follow, resulting in two cells. Each of these two cells now has half the number of chromosome pairs as the original cell, but each chromosome still consists of two sister chromatids. Now these two cells enter meiosis 2. There is usually no interface or DNA replication before meiosis 2, because the chromosomes are already duplicated, still as sister chromatids. Meiosis 2 is much more like mitosis. In Prophase 2, the chromosomes condense again if they uncoiled. In Metaphase 2, the chromosomes, each still made of two sister chromatids, line up individually in the middle of each of the two cells. Spindle fibers attach to the sister chromatids. This setup looks very similar to metaphase in mitosis, but it's happening in two cells simultaneously. Finally, in Anaphase 2, the sister chromatids are pulled apart. They move to opposite poles of their respective cells. Once separated, they're considered individual chromosomes. Telophase 2 and cytokinesis then occur in both cells. This results in a total of 4 new cells. Each of these 4 cells is haploid, meaning it has only one set of chromosomes, 23 in humans. And because of crossing over in Prophase 1 and the random way homologous pairs lined up in Metaphase 1, these 4 cells are genetically different from each other and from the original parent cell. This genetic variation is very important for evolution. Section 8, Mitosis versus Meiosis, spotting the differences. It is helpful to directly compare mitosis and meiosis to see how they're different. One major difference is their purpose. Mitosis is used for growth, repair, and asexual reproduction in some organisms. It produces new body cells, which are called somatic cells. Its goal is to make cells that are identical to the parent cell. Meiosis, on the other hand, has a very specific purpose: to produce gametes, or sex cells, sperm and eggs for sexual reproduction. Its goal is to make cells that have half the genetic material of the parent cell and are genetically different. Another key difference is the number of cell divisions. Mitosis involves only one round of cell division. It goes through prophase, metaphase, anaphase, and telophase once. This single division results in 2 daughter cells. Meiosis, however, involves 2 separate rounds of cell division, meiosis 1 and meiosis 2. Each of these rounds has its own prophase, metaphase, anaphase, and telophase. Because there are 2 divisions, meiosis results in 4 daughter cells, not just 2. This 2-step process is necessary to reduce the chromosome number correctly. The chromosome number in the daughter cells is a crucial distinction. In mitosis, the daughter cells are diploid, just like the parent cell. This means they have the same number of chromosomes as the original cell, for example, 46 in humans. The cells are genetically identical. In meiosis, the daughter cells are haploid. They have half the number of chromosomes as the original diploid parent cell, for example, 23 in humans. Furthermore, due to processes like crossing over, the daughter cells produced by meiosis are genetically different from the parent cell and from each other. This genetic variation is a hallmark of meiosis. Finally, let's consider where these processes happen. Mitosis occurs in most somatic cells throughout the body. It happens from the time an organism starts as a fertilized egg and continues throughout its life for growth and repair. For example, your skin cells, liver cells, and bone cells divide by mitosis. Meiosis, however, only occurs in specialized cells in the reproductive organs. In animals, this means in the testes for sperm production and ovaries for egg production. It does not happen in regular body cells. Understanding these differences helps us see the unique roles each type of cell division plays in life. Section 9. Why it all matters. The grand importance of cell division. Understanding cell division, both mitosis and meiosis, is incredibly important. It is fundamental to almost everything that happens in biology. Think about how you grew from a single fertilized egg into the person you are today. That entire process was driven by countless rounds of mitosis. Each time a cell divided, it created new cells that allowed your body to grow bigger, to form organs like your heart and brain, and to develop all your unique features. Without mitosis, there would be no growth from a baby to an adult. Life as we know it would not exist. Cell division is also essential for keeping us healthy. Our bodies are constantly replacing old or damaged cells. For example, the cells lining your stomach are replaced every few days. When you get a paper cut, new skin cells are made through mitosis to heal the wound. Red blood cells, which carry oxygen, only live for about 4 months and must be constantly replaced. All this repair and replacement relies on cells accurately dividing through mitosis. It is a continuous process that keeps our tissues and organs functioning properly throughout our lives. Meiosis too plays a vital role, specifically in the continuation of species through sexual reproduction. By creating haploid gametes, sperm and eggs, meiosis ensures that when fertilization occurs, the offspring receives the correct diploid number of chromosomes. Just as importantly, the genetic recombination and independent assortment of chromosomes during meiosis create genetic diversity. This means that offspring are genetically different from their parents and from their siblings, unless they are identical twins. This variation is the raw material for evolution, allowing populations to adapt to changing environments over time. Finally, studying cell division helps us understand and combat diseases. Cancer, for example, is essentially a disease of uncontrolled cell division. Cancer cells ignore the normal signals that tell them when to stop dividing. They divide too quickly and can spread to other parts of the body. By understanding the normal controls of cell division, scientists can develop treatments that target these runaway cancer cells. Research into the detailed mechanisms of mitosis and meiosis continues to provide insights into health, disease, heredity, and the very nature of life itself. It is a cornerstone of modern biology.